Good morning, everyone. Welcome in the fourth session of this unit. In this session, we are discussing about bioprocess technology. I am Dr. Arpita Das, Assistant Professor, Department of Biotechnology, Adamas University. And we were discussing about different aspects and application of bioprocess technology. In our last three sessions, we have discussed many points regarding bioprocess technology, and we are continuing about media formulation, and after that we are discussing about strain improvement. These 2 points comes under upstream processing. These 2 points comes under upstream processing. Okay, so let me share my slide. Okay, I hope the slide is visible to you all. Okay, let's stop sharing and share it once again. Okay. We are in this slide where we are discussing about different type of microbes, like different type of methods which are being taken as we are discussing about different methods in which we can improve strain. So some of them includes foreign DNA, some of them do not include foreign DNA. So strain development can be divided in two categories. One category where we use foreign DNA and another category where we do not use foreign DNA. So the category in which we use foreign DNA is a method involved recombination and genetic engineering or recombinant DNA technology. So transduction, conjugation, transformation comes under that area. The method in which we do not use foreign DNA is none other than mutation method, So optimizing environmental condition, as I have already discussed, that certain physical parameter like temperature, agitation, we need to optimize them to get a best product. We need to optimize them to get a best product. We need to modify the chemical parameter like certain pH, amount of oxygen concentration, that are to be taken care during media formulation as well as in bioprocessing. Some biological parameters like enzymes, addition of enzyme, how much amount of enzyme has to be added over there, that has to also be taken care. So these are the different aspects where we are supposed to optimize the environmental condition for the microbes to grow. And this comes under strain improvement. So we are talking in this session, we are talking about strain improvement initially. Now, during strain improvement, we need to take care that for that particular strain, that their nutritional resource has to be optimized. We have already discussed microbial media formulation, so that knowledge are supposed to be added over here for the perfect or optimization of nutrition for microorganisms. These are few list of genetically regarded as safe microorganism. That means in this microorganism, this improvement in their genetic structure has already been done, and they are being safely used by the human throughout the world. Under bacteria, Bacillus subtilis is widely used. Certain Lactobacillus bulgaricus, which are used for yogurt production, sourdough production, those are also been generally regarded as safe. These are proper strain. These are proper strain which are used in industry and which are genetically modified. Under that, we can talk about leuconostoc. Leuconostoc is again a microorganism which is used for fermentation of carbohydrate mainly. So for the various range in Earth includes different parts of European countries, different parts of Asian countries. They consume fermented cereals products. So for that, leuconostocs are being utilized in many areas. So lactobacillus or lactococcus is also being utilized for production of yogurt. Yogurt is widely used or consumed by the people. So in various countries, uh, like European countries as well as Asian countries, American countries, people consume yogurt because it's a good source of good bacteria. So these are the good bacteria which is inside it, that lactobacillus, lactococcus, leuconostocs, those are been there. Different yeast like Candida utilis, Culivoromyces. So those microorganisms, certain group of Saccharomyces cerevisiae, these yeasts are genetically modified to get a highest amount of yield. So these microorganisms are also being utilized. Certain fungi like Aspergillus niger, Aspergillus oryzae, certain mucor, Penicillium roqueforti for cheese production. So these microbes, this is not the not only microbe, but there are many. For your kind information, we have discussed few to remember. Like these are the few microorganisms which are genetically modified and widely used. So we have to remember at least few names from here. Now we are going to discuss those methods which are being adopted for strain improvement. So that includes mutagenesis. Yes, mutagenesis is a process or a treatment given to the microorganism and um, which will cause an improvement in their genotype as well as phenotype performance. So mutation can be spontaneous, it can be induced, and like mutation can be direct or indirect. That means mutation means the certain change in their genetic structure, certain change in the genetic structure of the microorganism. It can be imposed or it can be natural. Okay, so I hope that you have an idea regarding mutation in your genetics. Then also we are going to discuss about some uh, some type of mutation, like spontaneous mutation. What is spontaneous mutation? A mutation that arises naturally and it's not a result of exposure of certain mutagen. It's also called as natural mutation or spontaneous mutation. That like as an example we can say every day we are going out in the sun, we are present in the sun, everyone is not using that sun protection cream and all, so we go to the sun and the sun ray content UV ray. Yes, UV is one of the mutation causing element. So naturally when we go outside certain cells can get mutated. It's not likely, it can happen for every microorganism, for any microorganism it can happen. Okay, so regarding second one is induced mutation. Induced mutation is a method in which mutation is produced by treatment with physical or chemical agent that affect the DNA of the living microorganism. That means I am going to give certain component, either it is a physical or a chemical, and it will cause a mutation. Like suppose this is a microorganism and I am giving certain rays or certain light or certain chemical so that it has certain mutation. It can have certain mutation. Its characteristics get changed. So that kind of mutation is called induced mutation. And regarding induced mutation, the rate of mutation can be increased by various factors and agents called mutagen. So that means what is mutagen? Mutagen are the element which is the causating agent of mutation. That means this one is causing mutation. So this one is mutagen. Okay. Now, there are various types of mutagens. Okay, there are various types of mutagens are there, like X-ray, gamma ray, ultraviolet ray, various chemical like mustard gas, benzene, ethidium bromide. So many mutagens are there in environment, or we can use them, we can produce them to create certain mutation. So this is an example of direct mutation. You can see first example include point mutation. So I have pointed my cursor over here, point mutation or substitute mutation. That means you can see this is a normal DNA, and where in presence of certain mutation, what happened that the second this A, this A is been replaced with a T. A is been replaced, substitution of one DNA, dideoxyribonucleoside, by means of another. So it is causing T. So initially this in case of mRNA, UCA was being produced. Now after mutation, it is ACA is being produced, and the protein folding will be getting changed. Okay. Now, again, it is a deletion of mutation. You can see this is a deletion of mutation. This is a normal DNA. You can see normal DNA in a same position, a deletion. This deoxynucleoside has been removed. It's nothing but it is removed. So as it is removed, this triplet codon will be no longer working. So you can see initially it was UCA. Now there is one deletion to it so that it created CAC, it has been created. So this is a type of mutation we have discussed. Okay, another two examples are there. You can see here addition of nucleotide. Yes, initially this is AAT, AG. Initially that 5 nucleoside is this, and now it is being changed to, you can see addition has happened to in that insertion of one deoxynucleoside has been done. One G is been included over there, so the series has been changed. The reading frame has been changed, and this is called addition of this nucleoside. And the last one is a last one example, because we are not studying mutation, we are talking about strain improvement. So I'm not going to details of that, but the last example is substitution of nucleoside result in a wrong codon. So you can see here that the C is being changed with T, and initially from ACA, UGU is been produced with which refers cysteine, but here as the C is been replaced with T. So in mRNA you can see UAU. So we are supposed to get UGU, but I'm getting UAU, and we are supposed to get cysteine, but here we are getting tyrosine. So these are the example of mutation. In small mutation can change in the protein structure and protein folding, so which will create a difficulties, or which will create a change in the genetic structure. Okay, so these are the example where we are not adding any stretch of DNA. We are not adding any stretch of DNA. It is a process of changing or improving the microbial genetics by means of mutation. Next method where we are adding some foreign DNA, that includes recombination, transduction, transformation, and conjugation, along with certain genetic information like RDT or something like that. So we are going to talk about transduction. Transduction is a transfer of bacterial DNA from one bacterial cell to another by means of bacteriophage. So generally transduction is being done using two methods. One is called generalized transduction, and second one is called specialized transduction. If you share a photograph, it will be easy for us to understand regarding transduction. You can see here in photograph A, generalized transduction is taking place, and in group B, and in photograph B, you can see specialized transduction is being taking place, where a foreign DNA, you can see the blue part is foreign DNA, which is being incorporated into the microorganism. Here in same also in group B, specialized transduction, the blue part, which is a foreign DNA, which is being incorporated over the microorganism. So genetic improvement or strain improvement has been done using this methodology. Next methodology is transformation. Yes, you can see this is an easy diagrammatic representation of transformation. When a single stranded foreign DNA is absorbed by the ingredient with the genome and of the donor cell, like cells, the microorganism will absorb a single-stranded DNA from nature, and not all the microorganisms are being capable to take this natural degraded DNA from environment. The microorganism who are capable to do this is called competent cell. So that means that only competent microorganism can absorb this single-stranded DNA from environment. You can see here this bacterial recipient cell. I will put my cursor on the bacterial cell. That recipient cell has a chromosomal DNA with small a, b, c, d letter. Now, when this is a competent cell, when they came into contact with this fragmented DNA, single-stranded fragment DNA, this part is been this microorganism is capable to accept this single-stranded DNA into it, as it is a competent cell. You can see A and B, capital A and B part is been uh, came here into the microorganism, and this B part recombination has been occurred between the donor DNA and the recipient DNA. You can see this recombination has taken care, and the small b part get out from the genomic DNA, but the capital B part is incorporated over here. So this cell is genetically transformed or genetically modified cell, because you can see the small b letter has been replaced with the big B letter. So this is a transformation. So that means the foreign DNA part, this B section is foreign DNA part. This has been injected or inserted into this bacterial cell. So as an example, you can talk about bacillus species who are like naturally competent cell, and we can use bacillus in a huge manner for this situation. Now next part is, as we said, transduction, transformation, and conjugation. So the last one is conjugation, in which conjugation is involved cell to cell contact or through a sex pillars to transfer their genetic material, Okay, you can see this is a F plus cell. You can see this is a F plus cell. That means they have a fertility factor or F plasmid in it, whereas this is a F minus cell. That means it do not contain any this F plasmid. So both of them has created one conjugation pili, and using that conjugation pili by means of rolling circle model, that one first you can see the dark green section of the DNA get into this F minus cell, and they create its complementary stand over here. And after that, you can see all the cells, two cells. Initially, it was F plus, it has been F minus, but now after this conjugation is over, both of them became F plus. So this is a conjugation method. Like this cell was initially devoid of this F plasmid, but after this improvement, they have F plasmid in it, and F minus cell become F plus cell. So plasmid plays an important role in the formation of some industrial product, including many antibiotics. So antibiotic producing plasmid are being injected to some other bacteria to produce much amount of that antibiotic. Okay, so conjugation is one of the method which have been readily adopted for strain improvement. So without them, there are other various methods like genetic engineering, recombinant DNA technology that are also been taken care. Like we can see cutting of donor DNA, restriction endonuclease, which cuts DNA molecule and in specific site in desired fragment, cloning a gene. So that also been under genetic engineering. So these are the different method of strain improvement. But only strain improvement is not there. So when we are talking about uh, upstream processing, we have to take care few things. Like when we are dealing with microorganism, yes, you have genetically modified some microorganism. Good. Now you are supposed to use that for your production. That is also good. But then also I'll stop share the slide, and I'll continue that suppose you have created certain microorganism you have improved the strain, that is perfect. But now you are going to utilize it for production. Yes, regarding production, it is perfect, but during production, it can get contaminated. During production, it can get contaminated. So, what you are going to do? You are supposed to take a copy of that, or you are supposed to take the improved microorganism copy to keep somewhere. Okay, so that is called strain preservation. So, strain preservation also plays an important role. Strain preservation also plays an important role. Some culture collection type lab. We need to remember the word culture collection type laboratories are there where we generally keep the microorganism for future. Like in India, we have MTCC. There are many institutes where we keep the microorganism for future use. And okay, so I'm going to share my slide with you. Uh, In case of strain preservation, there are various type of preservation methods are there. Okay, but before going to preserve the strain, you have to understand what is the type of microorganism you are working with, what is the uh, uh, like how long you are preserving the microorganism. These plays also an important role. Like what is the type of microorganism, which is an important criteria. Like this is a type of microorganism, this type of microorganism I need to uh, score for, I need to save them for a long time, or how long I am saving the microorganism. Those criteria also plays certain important role. Okay, so preservation of strain is important. Now, just give me one moment. Okay, so this is something else. We are coming to this later. Just let me complete the part strain preservation. We are talking about culture collection type lab where microbes are being kept for a long time based on certain criteria, that which are what type of microorganism it is, like whether it is prokaryotes, whether it's eukaryotes, that thing. Second important thing, like for how long I am keeping them, for how long, it's for 3 months, 4 months, it is 5 year or 10 years. So these are the length of preservation. So these two criterias plays an important role while we choose type of preservation. Now, strain preservation or microbial improved strain preservation can be categorized in two categories. One is called short-term preservation, another one is called long-term preservation. Now, regarding short-term preservation, like in our practical lab or in our university, when we work with certain microorganism, what we do, we we buy those improved strain from certain type culture collection lab. In India, we use MTCC, Microbial Type Culture Collection Lab, which is in Chandigarh. We buy from there that microorganism and we break the vial, we culture the microbes in 2-3 petri plate, and we keep a stock of that, and we do serial subculturing. That means after 2 weeks or 3 weeks, when the media is been exhausted, no nutrients has been left in that plate, we discard that plate, and from there we take a small amount of microbes and we put it into another plate. So serially we do subculturing. So short-term microorganism preservation, short-term microorganism preservation is one of the important and like useful method where we do serial subculturing. Serial subculturing is nothing but after certain period, like 2 weeks, we do subculturing of the microorganism. Sometimes, uh, microorganism generally use oxygen. So some aerobic microorganism, they use oxygen for their growth. So we we put certain oil on the microorganism. Like we can take an example, like suppose I am having, this is a test tube, and in this test tube I have created a slant. In that slant, microbes are growing. Now we generally what we do, we put uh, we put a layer of oil on it. We put a layer of oil on it so that the absorption of oxygen is limited. Microorganism will grow, but it will get a lesser oxygen, so that their metabolism is little bit slow, and that is another method of preservation, short term preservation. Okay, that preservation, laying some unreactive, or you can say non-reactive oil on it. Paraffin oil sometimes been used. So these two are short-term preservation technique. One is serial subculturing and another one is serial subculturing, but layering the culture with certain non-reactive mineral oils. These are two short-term methods. Now there are two most important long-term preservation method, includes cryopreservation and lyophilization. Lyophilization means freezing under very low temperature. So lyophilization is a method which is called freezing under low temperature. That means microorganisms are being kept in a very low temperature for a long time. Microorganisms are kept in a very low temperature for a long time, so that they are metabologically uh, lesser active and they can stay there for a long time. And same thing when being done using liquid nitrogen, under liquid nitrogen, that is known as cryopreservation. So lyophilization and the cryopreservation are two most important method for long term preservation method. Now under upstream processing, we have studied media formulation. We have studied uh, that one is called media strain improvement. Yes, strain improvement we have studied. So these two things are important. Now, when we perform a practical in laboratory, initially we prepare the media, then we choose the strain, and then we go for media sterilization. That means we are supposed to remove all the microbes which are present in that media, because my desirable microorganism is supposed to grow on that media. If there are other microbes is there, they are going to utilize the food also. So what will be my aim? I will eliminate all the others and I will inoculate my desirable microorganism, and then only that will grow. That will be my aim. So uh, media sterilization or media and bioreactor sterilization plays an important role. So under upstream processing, we have studied media formulation, we have studied strain improvement, we have studied strain preservation. These three parts is important. And after that, another fourth unit or fourth section is important, which is called media as well as bioreactor sterilization. So fourth part is media as well as bioreactor sterilization. What is the definition of sterilization? Sterilization means complete removal of microorganism. Sterilization means complete removal of microorganism from that area or from that matter. There is no partial sterilization. Either I say this this pen is sterilized, that means no microorganism is present on it, or we can say it is non-sterilized, that means there is microorganism. There is no partial sterilization. We cannot use, yes, this is two microbes are sitting over there. No, there is no terms known as partial sterilization. Okay. So if the fermentation is contaminated, that means if there is other microorganisms, there is a huge loss. So we are not supposed to afford the loss in bioprocess technology. If the microorganism is contaminated, the medium would have to support the growth of both producer and contaminant microorganisms, resulting in the loss of productivity. Yes, correct. Number one point is correct. In case of continuous cultivation of the contaminant may outgrow the product organism. That means two microorganisms are there. One is my desirable, one is contaminant. Now contaminant has start growing so much so that productive organism, they are not getting their nutrition. So they will dry out or they will not grow much. So that is again a non-profitable condition. In case of biomass as product, the contaminant will result in a total loss of product. Suppose I have an industry where I am producing yeast cells. Now, while I am producing yeast cells, certain contamination has been started over there and some other microbes is growing along with yeast cell. So it will be very difficult for me at the end to separate them, and it will create a complete loss of the total product. So I cannot afford that. That means contamination has to be removed. The point is, I want to make you understand that there is no chance of contamination in a huge or large scale bioprocessing. It will result nothing but no profit, all loss will be there. So we cannot afford contamination. So fourth point, we can say the contaminant may produce compound which will make downstream processing difficult. Yes, along with this, my aim is to grow only this bacteria and this will produce certain protein. So along with this bacteria also has started growing and they will also produce certain products. Now all the products will mix together and it will be really difficult. It will be really difficult to remove them. So we cannot afford that. So we are supposed to remove the contamination by means of what? By means of sterilization. We have to opt sterilization very wisely so that if the sterilization method is perfect, then then the media as well as the bioreactor will be free from any microorganism. Now, for avoiding contaminants, what we can do? We can use pure inoculum to start the fermentation. We can use pure inoculum to start the fermentation. We need to sterilize the medium which has to be employed. We need to sterilize the fermenter without any fail. Sterilization of all the materials which are added in fermentation method are been taken care, and to maintain a aseptic condition during fermentation. These are the 5 thumb rule which we have to provide during this method, like pure inoculum which has no contamination, proper sterilization of the medium as well as bioreactor, sterilization of all the ancillary support of bioreactors, and to maintain a aseptic. That means aseptic means no other contamination will happen. There will be no hole in the bioreactor. There will be no such handling, careless handling must not be there. So these are the key area which we are supposed to take care. Now, pure inoculum we are supposed to produce using those strain improvement method. Next, sterilization of the media to be employed. Now, we are supposed to utilize certain method. It's not that autoclaving will work always. So, we are supposed to utilize certain method which include filtration, which include heating, X-ray, gamma ray, those kind of like ionic rays can be utilized as one of the uh, sterilizing agent. So not only physical, not only heat like that autoclaving will work, but we can use or we need to protect that fermentation from the microbes by various methods. I have listed few methods like how we can sterilize the media as well as the bioreactor. Like we can use filtration of the medium, we can use radiation over there, we can use ultrasonic treatment, certain chemical treatment, and the most popular heat treatment is there, which can be utilized to sterilize the medium, like nutrients, food for the microorganism. For the cleaning or sterilization of bioreactor, this radiation, heat, and chemical treatment plays an important role because bioreactor is a vessel. It's nothing but a huge vessel. So to clean that vessel, we can put certain UV lights in it. 10 UV lights we can put into it, and they will work for 1 hour, 2 hour, 3 hour, and the inner part gets sterilized. So that can be taken care. So media as well as bioreactor sterilization plays a very important role in upstream processing. Media sterilization can be done using filtration. We can use different type of filter like uh, absorbate or some different type of filter we can use. Regarding radiation, X-ray, gamma ray, we can utilize. Ultrasonic treatment, yes, ultrasonic treatment can be taken care. Different chemical, like for surface sterilization, we can use alcohol. Some other things are also there, some other chemicals are also there. We will study them separately. As well as heat, dry heat as well as moist heat under pressure can be utilized for media sterilization. So sterilization of media is one of the most important criteria under uh, upstream processing. So you can say, as I have already said, that X-ray has a lethal activity. X-ray has a very lethal activity against microorganism. So that has to be taken care. We can see chemical like different alcohols, different halogens, phenols and their compound that can also act as a good chemical sterilant for microorganism. So in this session we have discussed few things like regarding upstream processing plays an important role in bioprocess technology, and upstream processing include many steps. That all the steps has to be taken simultaneously, one by one, in a perfect proper manner. If there is any problem in any step, it will hamper the end product. So we are not supposed to we cannot afford that problem. So we have to be perfect in every step. Media formulation has to be done in a proper manner. It should be cheap so that microorganism can utilize it and they can produce the best product. Next, strain improvement has to be done in a proper manner so that strain can utilize whatever nutrient I am providing that. So strain improve, and utilizing that, it must produce the best amount of product, the best quality product. Third is like while I have done a strain improvement, that copy of strain has to be kept somewhere for a preservation, because maybe during the process contamination can occur. That time I need the copy once again. So strain preservation again plays an important role. And lastly, we have discussed about sterilization. The media as well as the bioreactor has to be sterilized in a proper manner so that the desirable product can get produced in a bulk amount. Okay, so these are under upstream processing. These are under upstream processing, and after a proper upstream processing, everything are being dumped or kept into a bioreactor. So bioreactor is a heart of bioprocess technology. Now in our next session, we are going to discuss about a little regarding bioreactor and how do we do production. We'll take an example of that. So until that, thank you everyone. Stay safe and well. Please go through the slide once again for your easy understanding. Thank you. Bye bye.